to do things. Okay, so my lab works on microRNA called microRNA 122. And it's an endogenously expressed small RNA that exists in cells. And what it does for HCV is it actually very potently promotes the virus replication. And it's been mentioned in a few talks already as a target for drug therapy. So we don't entirely understand how this microRNA works, and that's what my lab is working on doing, but that's not really what I'm going to talk about today. I'm talking about sort of strategies to target it as a therapy. And of course, microRNA 122, its job is not to promote HCV replication. It's actually, its real job is to regulate cholesterol biosynthesis, and it's also been characterized as a tumor suppressor. Being a very potent promoter of virus replication, anything that we as basic scientists identify, that becomes a good target for therapeutic intervention. And in fact, there are two companies that have been interested in using nucleic acid-based inhibitors to actually sequester miR-122 away. So basically bind it all up and make it unavailable for the virus. And these work actually very well. So one is miravirsin made by Centauris, and there's another one called RG101 by Regulus. And in clinical trials, these can be injected. They actually last a long time, and they can decrease viral load levels by several logs over a number of weeks. So one problem with the use of these siRNA, the miR-122 sequestration, is the concern about using them for a long period of time, since miR-122 is a possible tumor suppressor. So if you suppress it too much for too long, you could have tumors. So we came up with a strategy to spare miR-122 and actually target the miR-122 target sites. So they're very specific sites on the genome to which the microRNA binds. So just a very quick background on siRNA. So siRNAs are related to microRNAs, but we use them as scientists in the lab to knock down genes. Okay, so basically you can order a double-stranded RNA, you can order them online, they get delivered a few days, they're synthetically made, and you can then introduce these into cells. They will you'll choose a sequence that's perfectly identical to a sequence in a gene, and it will bind to that sequence with a protein called Argonaute, and it will induce cleavage. Okay, so very, very frequently used in basic labs. So here's a little animation about how we want this to work with HCV. So we've designed siRNAs that target the miR-122 target site. They're going to be incorporated into Argonaute. One of those strands gets selected. It binds to the 5' UTR, and it will induce cleavage. Okay, so one of our strategies, we know that knockdown relies on Argonaute 2, and miR-122 binding also relies on Argonaute 2. So we figured that target site should be a region that's accessible to both Argonaute and the siRNA, so it should be a good site. So our objectives were to design and test siRNAs that target that miR-122 binding site. And then with any sort of antiviral drug, you want to assess resistance. So we wanted to assess the potential of HCV to actually escape the siRNAs. And it can escape simply by introducing single nucleotide or nucleotide changes in the genome. The dogma is that those single nucleotide changes should abolish or at least attenuate the siRNA. While HCV can evolve, and we've seen a lot of information on resistance, since the miR-122 binding sites are actually are highly conserved, so those sequences are highly conserved, we know that they're essential. We actually first hypothesized that we shouldn't actually get very many mutants. And if we did get mutations, they could tell us something about the biology of HCV. And so if we do get mutations, we wanted to assess their fitness. So we designed these three siRNAs that are listed here, and these were designed using algorithms that will predict where siRNAs that work relatively well. And in a knockdown, a normal knockdown assay not involving HCV, we found that these two worked relatively well. When we looked at the knockdown in the context of HCV, we actually found that the first one, 18 to 36, still worked pretty well, so it knocked down about 95%. The second one, 19 to 37, wasn't quite as efficient. And I'm not going to talk about the third one because it didn't work all that well. So we conclude that siRNAs can target the miR-122 binding region and quite efficiently suppress HCV replication. So the next thing we wanted to do is assess the potential to escape siRNAs. And I don't have a whole lot of time today to go through the method, but it generally involves a repeated sort of treatment and selection for HCV that can survive. This is all done in cell culture, so we're selecting for viruses that have survived the knockdown. So this is with a control siRNA, and this is one we used years ago and showed that it could become resistant. So we could actually detect a decrease in efficacy after each round of treatment. So there was resistance and a lack of a decrease in efficacy. With these two siRNAs against miR-122, we actually didn't see that. We didn't see much of a significant decrease in the knockdown efficiency, suggesting that the virus maybe couldn't escape them. So the next thing we wanted to do was actually sequence. So we cloned and sequenced that 5' region, the region where miR-122 binds. And to our surprise, we actually found a great deal of mutations. So we sequenced just under 300. 210 of them had mutations somewhere, and 65, so about 20% of the genomes that we actually isolated, had mutations in the miR-122 sites. The previous slide was with the first one, that 18 to 36. With the other siRNA, that didn't work as well. We did still get escape mutations. Fewer of them, are about 12%, and a smaller distribution. And these are our controls. So in naive cells, we didn't have any mutations in that region, and just in selection with the siRNA that targeted somewhere else, we had a few sort of changes, which are probably just growth adaptation mutations. So the third objective we've got is to assess the replication fitness and the siRNA resistance. So are these things actually resistant to these siRNAs? So thus far, we've cloned 35 individual unique mutations that we isolated. And this is a work in progress. So I'm going to give you just a summary of what we found out so far in looking at some of these mutations that we selected, and also one mutation that we actually use quite frequently in the lab that we didn't actually select in this. So the summary is that inhibition, so we did see what we expected, that some of the point mutations had, I'll go back one slide, sorry. Most of the viruses actually appear viable. Okay, so they can replicate. We haven't looked at particle formation, but the RNA can replicate, but generally they're less fit than wild type. So we did see what we expected, that some of the point mutations, the siRNA knockdown ability had been attenuated. For some mutants, the actual knockdown was actually enhanced on the mutant rather than the wild-type virus, which was surprising to us. And we still don't entirely know the mechanism by which that's enhanced. And then interestingly, we actually found that the siRNAs could actually turn into miR-122 mimics in a couple of cases. In one case, it's quite a poor mimic, and in another case, we actually have quite a strong mimic through just not very many sequence mutations. So we think that the mechanism that these siRNAs are working could be manyfold. So this is the standard method. It's going to bind and cleave. Even in the presence of a mutation, it may still be able to do that. It may be able to displace viral proteins from that region of the genome, or even displace miR-122 from that region. We also think that some of these siRNA mimics, at least if they're poor ones, would then compete with miR-122, miR-122 being a good one. The siRNAs being a poor one, you would actually get an inhibition through that competition. So in conclusion, we have siRNAs that can target the miR-122 binding region, and they do inhibit HCV. Escape mutants do evolve, but may not necessarily abolish inhibition. And siRNAs can become miR-122 mimics on mutant HCV genomes. Thank you. Oh, one more. One more. Sorry. Just the last
question I can.
Sorry, I forgot about acknowledgements. He won't let me through. Oh, okay, never mind.
Hi, Joyce.
Adam Hayes did this work, so.
Great talk. I was just wondering if you made a oligonucleotide that was essentially microRNA 122 without any of the bulge, so it was completely complementary to the region, what happens?
I'm not sure we've made that one. I think we tried to go through the longest one, the one that didn't work. Actually, we tried to go through both of the seed regions, and it was actually one on the algorithms that wasn't predicted to work very well, and it didn't. Did we make a miR-122 that just sits down? One of them is similar to that, but not quite. So no, we haven't.
Okay.
Do you know if the mutants bind to MI?
I'm sorry?
If the mutants bind to miR-122?
If the mutant viruses bind to miR-122?
The mutant sequences, at least.
With the mutant sequence? I'd have to ask Adam. We haven't looked at very many of the mutants yet, but we are looking at them in the context of sort of with and without miR-122, and we should be able to see if they bind.